Thanks for coming. Pleasure to introduce our next speaker in the seminar series, Dr. Rich Smith from the University of New Hampshire, coming to us. He's an associate professor of agroecology in the Department of Natural Resources and the Environment at the University of New Hampshire. He's been there since 2010.
Yep.
And before that, Rich did several postdocs. The most recent was at Penn State, and a couple others with Montana State, and I think you did one with USDA ARS in the Bay Area. Got his PhD at Michigan State and a bachelor's degree from the University of New Mexico. And we're glad to have him here. And I'm not going to try to read his title, so I'll let you.
Yeah. Thanks, Ben. Thanks for the invitation. It's been really fun to visit this campus. I hadn't been here before and interact with the grad students and the faculty, and so I'm very appreciative that you invited me. And this title is really pretentious, but I was trying to play off of. Hopefully, you get the Wizard of Oz reference. So. All right, so just brief outline. I just want to do a couple things here. I want to give you just a flavor for agriculture in New England, because that kind of sets the stage for the research that I do. There's some interesting emerging initiatives that have really kind of driven my research ideas and trajectories. And then I'm just going to give you 4 highlights from 4 projects. We have a bunch of things going on, but I tried to pick 4 that I've been thinking about recently. So I'm not going to go into a whole lot of detail on any of those 4. Some of them more so than others, but hopefully they'll give you a sense for the types of things we have going on. So let's think about New England. So I'm gonna give you a bunch of these maps here. They're basically data maps. You can see I've circled the region New England there, and you can see Virginia for comparison. We grow a lot of forage crops in New England, which might be somewhat surprising. A lot of those forage crops supply our dairy, so you can see we're actually a pretty decent dairy region in the United States. Obviously, we grow a lot of vegetables, so that's, a lot of times people think about New England and they see, small scale diversified vegetables farms, and certainly we have a lot of those. I stuck Virginia there on the bottom too for comparison. So we're a bunch of very small states, right? 6 very small states, but we have a lot of vegetable farms as well. A lot of our farms are organic, so there's a lot of organic in New England. This is color-coded by number of organic farms, so sheer number, and we're a small state, so that doesn't maybe look real impressive, but if that was color-coded by percentage of farms that are organic, we'd look even darker blue there. What's really neat is that New England is one of the epicenters for local food. There's a lot of interest around local food. One blue dot is $200,000 worth in value of products sold directly to individuals, so direct sales. In fact, the 6 states of New England are in the top 10 of all states in terms of direct sales to customers, right? So there's a lot of local food, a lot of interest in local food. We're not a big food exporting region. So these are things that make New England a pretty unique and interesting place to do the type of research that I do. And there's some interesting initiatives that are kind of afoot that make it even more interesting, at least for me, and the types of things I'm interested in. The big one in my mind is this food vision. This food vision is something that folks in New England are kind of rallying behind. It's grown out from collaborations between the regional universities and a host of other stakeholders involved in the food system. And it's really kind of a bold vision for the future of agriculture in New England. And I've highlighted here this statement here that really kind of summarizes it, that a New England food vision calls for our region to reach a bold goal of 50 by 60. It's even trademarked, 50 by 60, building the capacity to produce at least 50% of clean, fair, just, and accessible food for all New Englanders by 2060. And this is a real thing. folks are talking about this at all levels, policy within the region. So there's this push to grow more food in New England, produce more of our own calories in New England. And you can download this, you can go on the website and check it out, and there's a lot of modeling involved and a lot of folks that have contributed to this. So that's one really interesting initiative, and that's really gotten me thinking about what does the future of agriculture look like in New England. The other thing is climate change, right? That's the elephant in the room. Here's some interesting model data showing water supplies across the country. Obviously, red and dark red are bad. Light colors are better in terms of water supply. This map here is just based on projected human population and kind of status quo water use. And then when you layer on top of that climate change, anticipated impacts of climate change, well, our region actually starts to look really good from a water perspective. You wouldn't know that this year. This year we're in the midst of one of the worst droughts we've ever had, and farms have gone out of business as a result. But in general, we're supposed to look pretty good for water. We need water for agriculture. So, you can connect the dots there. We have a push for agriculture. We might look pretty good for water in terms of agriculture. So there's some interesting things to say, and that is, agriculture is strong. Folks think about New England, they don't necessarily think about agriculture. It's strong and it's growing. It's growing and it's projected to grow even more. There's a lot of interest from stakeholders in expanding agriculture in our region. And then there's these external drivers like climate change, which may reinforce this expansion, right? It might be better and make more sense to do more agriculture in our region because we're going to have decent water supplies, right? So that's the state of agriculture, and there's some questions that emerge, right? If that's the case, then, well, one of the things we need to ask ourselves is how realistic is this vision, this 50 by 60 vision, right? 2, what would be the impacts of expanding agriculture to that extent? And you'll see it would actually represent quite an expansion to get to 50% by 2060, right? What would be the impacts in terms of environmental quality, all the things that we also need to be concerned about with regards to agriculture? And then how can we do it without screwing things up, right? How can we do it in the most kind of sustainable and responsible way? And that's really kind of driven my research program so far. So let's just quickly touch on these. The realism, how realistic is it? Well, first you need to understand what our current capacity is. Like, how much land do we have in agriculture? How much local can we produce currently? And there's a lot of different ways to do this, and this is one way using one set of data, but I think it's pretty accurate. You can see in terms of current capacity, it ranges anywhere from the low end of 3% in Rhode Island up to a high of 39% in Maine. In terms of capacity, in total across the 6-state region, 10% currently. We have the capacity to supply 10% of our agricultural demand locally, food production demand locally, right? So we're at 10% now. We want to get to 50 or maybe more, right? So let's think about what is the future then? What is the future capacity? Is that even possible? And this is just an example just for New Hampshire. So that's the state of New Hampshire there. And we can look at our current acreage in terms of cropland, right? So 98,000 as of 2012. We can look at the prime agricultural soils, right? So we have just about double that in terms of prime soils that could potentially support agriculture. Some of those are probably under housing developments or otherwise unavailable, but not all of that for sure. And if we add on to that the state and locally important farmland soils, then we have over 1 million acres of soils potentially useful and appropriate for agriculture. So our capacity for future productions probably a lot greater than what we're currently at, right? So it's possible, right? It's possible that we could get up to 50%. What would be the impacts of that then? So let's assume we could do that. Do we want to do that, right? That's the next question. Is that a good thing? Well, one of the defining features of New England, if you've ever visited or seen pictures, is our forest cover, right? In about 3 weeks, the leaf peepers are going to be invading our region to see our deciduous trees turning colors. In fact, New Hampshire ranks #1 in the United States in terms of percent cover. We're at 88%, 89%, right? And all of our 6 states are in the top 9 in terms of percent cover. So we have a lot of forest, right? And that forest is a net carbon sink right now. So we're storing a lot of carbon in our forest. So from a carbon perspective, what's that mean to change to a lot of agriculture and necessarily less forest, right? From a water quality perspective, what does that mean to remove some forest and replace that with agricultural lands? So these are the types of questions that have gotten me very interested. But there is a precedent for a greatly expanded agriculture, right? If you go back 100 years, 150 years ago, the landscape of New England looked very, very different than it does right now. So here's time on the x-axis. And, in the mid-1800s, early 1900s, percent cover of forest in places like Massachusetts, Connecticut, we're talking like 30%. 70% of the area of those states was in cover other than forest, so grassland, other forms of agriculture, right? And then as agriculture moved west, a lot of that land was abandoned and a lot of that land was reforested, right? So we're up here now, contemporary, and there's been actually some increase in the forest cover since 2000, since these data captured. And in fact, if you walk around in the forests of New England, you can see the remnants of our agricultural past. You'll be in what looks like old growth forest. It's all mostly second growth forest, but you'll find stone walls crisscrossing these otherwise impenetrable forests. And those stone walls in the day separated adjacent pastures, probably sheep pastures or other livestock. There was a lot of sheep back in that time. But you see the reminders of our agricultural past very clearly throughout the forests of New England. So there's a precedent. It's happened in the past. Right? So here's what we know. We're practicing agriculture on just a fraction of our potential agricultural soils. Much of our ag soils are now forested. There's a historical precedent for a much larger scale forest to agriculture conversion. I mean, that happened a long time ago. Nobody monitored it. Nobody measured what happened when all that happened. And we have these local and regional initiatives that are calling for an increase in our local agriculture from 10 to as much as 50 or even more percent. And so the third piece of this is how might we accomplish these goals of agricultural expansion in the most kind of sustainable way. And so, we can think about these as 2 sets of strategies, right? Intensification strategies, so that's got to be part of it. Using the available land, the land that we already have in agriculture, using it to be more productive, right? So intensifying our production on that land already. But there are going to be some extensification, right? We're going to have to grow more agricultural land, and how can we do that sustainably? So I have a number of projects that are looking at different aspects of these 2 questions, mainly on the sustainable intensification side, and I just want to give you some highlights from 4 of those. And so the first kind of experiment that I want to talk about is some work that we've been doing looking at genotypic diversity or cultivar diversity, and using cultivar diversity in our forage systems to gain some benefits in terms of productivity and other agronomic goals. And so what I'm talking about here, just so you know, is taking a stand of forage, so orchard grass, or in this case perennial ryegrass or alfalfa, and instead of planting seeds from a single cultivar, maybe we've got 100 seeds in this picture, instead planting 100 seeds that are made up of 2 cultivars, or 3 cultivars, or 8 cultivars, right? So that's what we're talking about in terms of increasing cultivar diversity, but not adding more seeds total. So keeping kind of the amount of seed constant, but just changing the proportions of cultivars that are represented. And so the conceptual model that kind of underpins this line of research that we've started and that we've actually kind of started to finish up is under a kind of a traditional single cultivar strategy, right? You can imagine that you've got an individual genotype that's been bred under some conditions, and it has some optimal environmental conditions where it performs optimally, and then if the conditions are less than optimal, it performs a little less optimally on either side, and that kind of defines the growing season of that particular cultivar. Right? And so if conditions are optimal, then productivity is going to be high. You're going to realize your agronomic potential in that cultivar. But we know that growing conditions are not static, right? And so in some years, growing conditions are going to be really poor, right? And so productivity is going to be poor. And as the climate becomes more variable, potentially, you're going to see a lot of variability in production. Now contrast that with a system where instead of planting a single cultivar, you've planted 3 cultivars, each with a different, environmental optimum, right? Hopefully very different optimums. You're potentially expanding that growing season, and maybe even more importantly, you are reducing the potential for variability with weather variability, right? So that's kind of the theoretical context for this. And it's born out of diversity ecosystem function literature and the idea that these cultivars should have complementary resource use strategies and facilitation and all of that stuff, that kind of underpins that body of theory. So that's the conceptual model that we're dealing with. And we're working with perennial ryegrass, so all this is in perennial ryegrass, but it should be transferable to any forage species or other crop species as well. So there should be a couple different benefits. In the intermediate, relatively short term, we would expect increased forage productivity throughout the growing season, potentially reduce susceptibility to invasion by weeds, because for the same reasons you might see that with species diverse plant communities. And then over the longer term, we would expect greater stability in forage productivity through time from year to year. So we've done 3 experiments around this theme, all at our research stations around the UNH campus. The first experiment we set up involved mixtures of 1, 3, and 6 cultivars, really kind of a classic diversity function experiment. The results of that were published in 2014, so I'm not going to go into those. You can read the paper if you're interested. Then we had a larger study where we basically asked, are there more benefits with more cultivars? Because in the first study we saw some productivity benefits by mixing cultivars. And is there a ceiling to that? Can you just keep on mixing cultivars and get even more benefits? So this other study is going from 1 to as many as 10 cultivars in a mixture. And those data we have yet to analyze. But the study I want to talk about today is where we're really taking a more concerted approach to creating these mixtures. Rather than haphazard mixtures, we're asking if we can create designer mixtures based on maximizing the range of traits. Right? And so this is funded by the USDA OREI program, so it's basically an organic research program. So this is all in the context of organic dairy. We've got a couple collaborators, so that the experiment I'm going to tell you about was replicated in Vermont, in Maine, and in central Pennsylvania, as well as at our site. I'm just going to be showing you the data from our site. Here's what we did. The treatments were created based on 2 traits. So we were lucky enough to get a bunch of cultivars of perennial ryegrass from Barenbrug, which is a large forage breeding company. They provided us information about the traits of these species in terms of their winter hardiness. Sorry, species, cultivars. right? We're dealing with cultivars, single species, dealing with cultivars. So we had information about the winter hardiness of each cultivar and the heading date, like time to relative maturity of each cultivar. So we were able to take that information and then create mixtures aimed at extending the grazing season, right? Because we have a very short growing season. So if we can extend the period of time that animals could be on pasture, that would be a good thing. Or that we can increase the productivity from of haylage or whatever, right? So that's the idea. So the first treatment, so we call it T1, represents kind of our control, kind of the standard approach, right? Single cultivar approach where you have a single cultivar, it has some range in heading date and some range in winter hardiness, right? And so hopefully this picture will illustrate how these treatments differ. Keeping in mind that all of the mixtures I'm about to show you have the same total seeding rate, right? So this is kind of the conventional monoculture approach. The other treatments expand the range of either heading dates, right? So keeping that same cultivar in the mix and then adding cultivars that have later or earlier heading dates. Or they expand the range of winter hardiness with higher or lower winter hardiness, right? So this is the treatment really aimed at season extension potentially. Or we expand both of those traits. Right? So those are the treatments, T1 through T7. Here's a table that just basically lays that out. So here's the heading date range, here's the winter hardiness range of the mixture, and then we can calculate something called the total trait range where you just add those two ranges up. Here's the number of total number of cultivars in that mixture, and then whether they're diploid or tetraploid or other information. And this is how you calculate the range, right? So that's an example. So if you have cultivars that are rated at 4, 5, 5, 6, and 7, the range would be 3. So that's how this is calculated. And you can see that obviously in that kind of control single cultivar, there is no range, right? It is just what it is. T2, we've expanded the heading range. We've also expanded the winter hardiness range. So for a total trait range of 3. There's 3 cultivars in that mixture. This one has 5 cultivars. We've really expanded the heading range and the winter hardiness range. Total range is 5, etc., etc. The point being here is that some of these mixtures expand both traits, and then other mixtures expand only one or the other, and then we've got some of the other potential variables covered as well. The treatments with the stars are the ones that contain that single cultivar. It was Remington was the cultivar, if you're familiar with it. Some of them don't. And then there was this commercial blend, right? So this is kind of the other control. You can actually buy commercial blends, right? A blend is just another word for a cultivar mixture. The thing about the blends is that they vary from one year to the next. It's basically what they have left over from the last year. They mix that up, and that's your particular blend for that year. And so the composition of the blend can change from year to year. So that's why I don't have any information about it. All right, so I think that's all I want to say about that. So in terms of methods, all of this was established in 2012. These are perennials, so it was a one-time establishment. Ryegrass seeding rates held constant. 5 replications at each site of each treatment. We also planted each cultivar in monoculture, which actually became pretty important, just so we could examine each individual cultivar that made up all those mixture treatments. We sampled ryegrass biomass from all treatments when they reached a certain height threshold, right? So they were all sampled at the same time. And that resulted in 11 total harvests from 2013 to 2015. Every time we harvested, we sorted the ryegrass. We also planted white clover with all of these as a companion crop. It was the same cultivar of white clover, same density, so that's not really a treatment variable. Looked at weeds, then we dried and weighed it. So those are the methods. And getting into the results, these are the 7 treatments just averaged across all the treatments, just to look at the trends in productivity at this site. And so you can see we had 5 harvests in 2013. We had 4 harvests in 2014. You can see something happened here, right? Something awful And then we had 2 harvests in 2015. So we captured that kind of early and late season productivity in the summer slump, right, when it gets hot and dry and those cool season grasses don't produce as much. Here's the mean yields for each individual cultivar in monoculture. And I show you this so that you can see, and these are the different cultivars. You can see things were going pretty well first year. And then a bunch of these cultivars really kind of petered out. And then things got real variable in the third year here. So consequently, I don't have a lot of confidence in these last 2 years of data. So the first data I'm going to show you are from 2013, where I feel pretty darn confident that all the cultivars that we included in the mix remained in the mix throughout the year. The great thing about giving seminars is it forces you to analyze your data. So I analyzed these data last week. All right. I analyzed them the best I could in the time I had. Not thorough, but enough to get a sense for what's going on. So here's total productivity over those 5 harvests of that year, broken out by treatment, right? There's the treatments, dry matter. This is sum of 5 harvests. So total productivity over the growing season, and sadly, no differences, right? So that's kind of a bummer. Bummer number 1. Bummer number 2, stability, right? So that's another thing we want to look at is how variable over time. So albeit we're talking about the variability in productivity throughout the season. So over the course of these 5 harvests in 2013. And again, so in this case, we're looking at the coefficient of variation, right? So a higher CV is more variable, lower CV would be less variable. There's some interesting looking trends, but bummer number 2, no differences there. But what I think is really useful is to look at both of those things, right? Because we don't just care about productivity. We don't just care about stability, right? It would be kind of crummy to have a very low productivity system, but it's very stable, right? That's not very useful. So we want to look at both of those things at the same time, right? And so this is one way to do that by plotting the variability versus the dry matter. And so as these numbers get bigger on the x-axis, that's higher productivity, that's better. As the numbers get bigger on the y-axis, that's higher variability or lower stability, that's not so good, right? So we basically create these 2 zones, right? The no man's zone of lower productivity and lower stability, that's not good. And then, you know, the sweet spot, higher productivity and higher stability. So that's really where we'd like to see these systems be. So then we can place our data in those quadrats, and you can see, again, keeping in mind that productivity was not significantly different, stability was not significantly different. So this is kind of an integrated measure. But it is noteworthy, I would say, that 2 of the 3 treatments where we expanded the range of both traits fall out pretty much in this kind of sweet spot area. And the treatment where we did not expand any of the traits is in that no man's land. So I'll leave it at that. You can form your own conclusions. I did throw in the rest of the data because we had the data. So this is now the sum of 11 harvests. There's some interesting hints at a trend here, but again, no statistically significant differences. Stability, same story. We're on bummer number 4 at this point, I think. But, okay, so was there anything different? Invasibility or weed abundance was different, right? So between T1 and T6, T6 was that conventional blend, the commercial blend, that had more weeds in it than the other kind of control, the single. So not too exciting there, and it roughly follows that trend in productivity, so it's probably more of a productivity suppression thing, and the p-value is 0.06, so take that for what it's worth. We can do the same type of productivity versus stability, and Interestingly here, now all three of the treatments that expanded both trait ranges are in that sweet spot, but interestingly, so is T1, right? So we'll see. There needs to be a lot more analysis of these data. This is just the New Hampshire site. We've got 3 other sites where this happened, so I think pulling in all that data, doing some more sophisticated analyses, I'm still hopeful that there's some interesting things going on here. So based on just these data, there's little evidence that these particular mixtures, which are very specific mixtures, have productivity or stability benefits over the single cultivar mix. I think part of that is because we use perennial ryegrass. I had championed orchard grass as our model system, and I got overruled by a collaborator, and I'm blaming it on that collaborator. I think orchard grass would have persisted. Perennial ryegrass had some persistence issues, and I think that screwed us up. It does appear that mixtures that expand the range of more than one trait may have some desirable features, especially if we analyze them with some sort of integrated metric, right? This CV versus productivity. But again, we need additional analyses. Staying with forages, new project. This is, we call it the silvopasture project. We're asking basically, can we expand agriculture while making use of the fact that a lot of our land is forested, right? If you're not familiar with silvopasture, it's an agroforestry practice. It's the integration of trees and forages and livestock, the deliberate integration of those things. So why silvopasture? Why New England? Well, hopefully you've gotten a sense, right? We're ripe for silvopasture, right? If you look at the distribution of prime soils or the distribution of prime and local soils, New Hampshire is covered with them. It's also covered in forests. So these are the prime and local soils that are under forested cover right now. So those are potential sites where maybe something like silvopasture or agroforestry might be a way to achieve some of our agricultural goals without wholesale clear cutting of our forests. Another factor is this: the land that is in farms in New England. A large fraction of that is forested. So you go to any one farm, they are cultivating just a small percentage of their farmland. The rest of it is forested. So that's the land, that's the forest that's already on these farms is probably going to be the first to go, right? These farms are trying to expand their acreage. What are you going to do? Well, you're going to cut some of your forest, or you're going to convert some of your forest to silvopasture. So we're interested in what happens when you do that. What are the agroecological and biogeochemical, so the carbon cycle, nitrogen cycle, water cycle implications of doing that. So we've just recently set up an experiment at our organic research farm where we were given some parcel of the forested land. We've got a forest control here. So this is the kind of native forest there, and we've either clear cut it, right, or we've cleared 70% of it, and this is our silvopasture. We've got mature open pasture that's been in pasture for at least 10 years, 20 years, and we're measuring a whole bunch of stuff. We have some carbon and nitrogen, greenhouse gas, hydrology, forage, timber, plant invasion. These are just some pictures from a little while ago. This is what the silvopasture looks like. You can see we've got all kinds of sensors up. This is the open pasture. We have a lot of slash. So a lot of slash from the harvesting process, which has basically created this woody mulch. So it's making it very difficult to establish forages. So we're on the very front end of this experiment, and we're working to get these systems up and running, but we're going to have a whole bunch of data about what happens when you do that initial clearing or that initial thinning to create silvopasture. Here's just some data. I'm not even going to go into it, but various data that we've already collected about through fall, so that's just water reaching the soil surface, saturated flow rates, so the roots in the forest are helping water percolate and infiltrate, and we've lost some of that in the silvopasture, but not to the extent that we did in the open pasture, and it depends on the depth as well. And we're partnering with a number of farms across the region where we're making these same measurements, where these stars are. So we're coupling our site with a bunch of other sites, some of which are replicated. So hopefully over the next couple of years, we'll have a much better sense of what could happen. Do these systems make sense for our region? There's very little data in our region. There's a lot more in this region. And so that's all I want to say about that. Switching topics because I'm trying to stay aware of the time. I passed this sign on my way to home. It was at a church, and I passed it, and I was like, oh, you know what? If I don't turn around and take a picture, I'm gonna kick myself. And so we're gonna talk about weeds now, briefly. So the third project I want to talk about was a collaboration between us, University of Maine, University of Vermont. Had a number of objectives. One was to basically categorize the weed communities across northern New England, so Vermont, New Hampshire, Maine, focusing on the soil seed bank. We were interested in understanding what correlates with the weed communities, what's driving the weed community variation across this region, and assess whether it's things like climate-related variables or soil-related variables that seem to be the major kind of determinants of the weed community. And then we had some other more molecular objectives. So I'm just going to touch on these. So the context for this work is that we know that climate change is going to bring with it changes in precipitation and temperature. And we know that we are seeing, and we certainly expect, expansions in ranges, shifts in ranges of other organisms. There's some projected changes in the ranges of different forest types with climate change. Everything's basically moving north, right? And we wanted to know if those same types of dynamics might happen in the weed communities in our region, right? Might we find new weeds, new problem weeds? Maybe some weeds become less problematic because they're no longer suited to the climate of the new region. Can we predict that based on the species? Can we use northern New England as kind of a canary in the coal mine? And I'll show you why, based on this high environmental heterogeneity, right? So if we think about something like the plant hardiness zone, right? This kind of maps on the environmental heterogeneity in our region. You can see that in our region, you can go 6 or 7 zones in the space of a couple hundred miles. Whereas in other regions, you would have to travel for days to cross that number of plant hardiness zones. So it makes for an interesting test case because we can hit so many climate zones in such a small spatial area, right? You can go from zone 6A all the way to 3A in a couple hours drive. So the question is, can we detect some sort of climate signal in the weed communities, focusing on organic farms, because organic farms should have the highest weed populations and highest weed diversity. And there's actually reasons to think that the answer to that would be no, that we wouldn't detect a climate signal. And that's because weeds are ubiquitous, right? These are some of the common weeds in New Hampshire, right? Lamb's quarters, barnyard grass, Canada thistle, broadleaf plantain, certainly dandelion. Do you have those here in Virginia? Of course you do. Anyone want to guess where I took those pictures? Close. Paris, last year, right? I saw those same weeds in Paris. I saw those same weeds in Czech Republic. I saw those same weeds in China, right? They're ubiquitous. So maybe there wouldn't be a climate signal, right? So there's reason to think that they're just kind of, there are multi-purpose genotypes. They are just there. We're not going to see anything. So in terms of methods, we sampled 77 organic farms. This happened in 2013. There's the number from each state. At each farm, we sampled the weediest field. The farmer told us, that's my weediest field. We sampled that. We sent a subsample to University of Maine for soil physical and chemical. And then the rest of it, we assayed for the seed bank community composition using direct germination. Yada, yada, yada. There's the locations. You can see we tried to cover as much of that environmental gradient as possible. I think we did a pretty good job. This is what it looked like in the greenhouse. So lots of weed seedlings identified and removed from these soil samples that were spread out and watered for months and months. We created 2 data matrices, right? The weed community matrix, basically the seedling density data from that seed bank assay from all the farms. And then we also create a matrix of these potential correlates, right? Our soil and chemical data, and then geographic data from each farm, and weather station data located in each farm, and those are the variables there. So basically we're saying, do soils or weather predict weed communities, or correlate with weed communities, I should say. Did a bunch of different things, including ordination. The major reason we ordinated the weed communities was to basically capture that variation in the weed communities across all those farms onto 2 or 3 synthetic variables that we could then look at those variables in relation to all of our other data on soils and weather. We did some regression analyses, and we did it at 2 scales, the regional, and then we broke the data set by state as well, and you'll see why in a minute. So we counted a lot of seedlings, right? 52,000 seedlings that represented 113 unique species. Some of the farms had really low seed densities, weed seed densities in the soil. Some of them had really high weed seed densities. So very, what you'd expect on a bunch of organic vegetable farms, basically. This is the ordination of the weed communities for the whole region, color-coded by state. Right? These are the weed communities. Each individual symbol is a farm or a weed community on a farm. And the first thing that jumps out at you is, holy mackerel, the weed communities in Maine are completely different from the weed communities in New Hampshire, and they're completely different from the weed communities in Vermont. That might be the case, but I kind of am a little suspicious, right? One of the things I didn't tell you is that each person in Maine and Vermont and New Hampshire did our own soil sampling. So we might have been somewhat different in how we sampled the seed bank, yada, yada, yada. But okay, let's assume that that has nothing to do with it, right? So we can look at the correlates. So this is the ordination axes, right, that now capture all that variation in the weed communities. And this is the Pearson correlations. I've broken out the variables by these kind of geoclimatic variables or the edaphic variables. So those are your soil chemistry, physical property variables. And any variable that had an R squared of 0.2 or greater is plotted here. And then the strength of that correlation determines the length of the bar. And they're in red, right? And so you can see for the regional level, we've got 4 variables that correlated with the weed community. Strongest by far was longitude, which makes sense if you think about how the states were laid out in that ordination. There were a couple edaphic variables, right? Lower correlation. So we had 3 edaphic, 4 geoclimatic, and the strongest being longitude, which is all obviously correlated with a lot of these variables as well, right? So these are just single correlations, right? Not taking into account correlations among these variables. So kind of informative, but not super informative. We can break it out and just look at state by state, because now I have a lot more confidence in these data, and I'm going to move along quickly. But the main point here is there's a lot more geoclimatic variables than edaphic variables in Maine. New Hampshire, same story. Vermont, even more so, right? None of these edaphic variables. We can apply some more sophisticated analyses that take into account the correlations among our predictor variables. This is basically showing which of those are most important for building the model that explains the community composition. These are loading scores, so the factors are now, these models are basically condensed into factors. The factors are influenced by these different variables. The first factor is far and away influenced by climate-related variables, right? The second factor, also climate-related variables, but to some extent are soil variables. But this is factor 2, which explains even less. So climate seems to be really important. Here's for New Hampshire, same analysis. And again, we explain a lot more of our community based on temperature variables. Just a couple soil variables come in, but mainly climate and geographic variables. I won't beat that horse, right? So Same thing. The soil variables don't become important until they explain a lot less of the variation, we'll say. So summary, at the region level, which I have medium confidence in, geographic and weather variables are the strongest correlates. At the state level, there's very little evidence that soil or edaphic factors are strong and or consistent correlates. It's all of these geoclimatic variables that seem to be explaining the weed communities. So, the takeaway is change in climate is likely to lead to change in our weed communities. Then the question is, well, which species are likely to be shifting in and out, right? Which are going to become more important or less important? We can look at this based on climate hardiness zones, because I think hardiness zones kind of integrates a lot of these climate and geoclimatic variables in one single metric, right? And we can use indicator species analysis. I won't talk about how that works. But basically, we can identify a number of species that seem to be strongly associated with particular plant hardiness zones. And so as climate changes and those zones tend to move further north, we can expect that at least some of those species are also going to either become more abundant in a given location or less abundant. I'll leave it at that. I'm not going to go over that. And I think I'm going to make it. I'm going to go through this real fast. This is the fourth project. This is a fun one because I got some imagery that I wanted to incorporate. So we'll go through real quick. This is a trading tillage for tarps to increase no-till organic. So organic no-till, if you're familiar, considered the holy grail of organic, right? Reduced soil disturbance has a lot of benefits, but you need that disturbance to control weeds. So until now, it's been very difficult to make this system work. So the way you make it work is you crimp cover crops to create a high residue mulch. I bet Stephen Mirsky talked about some of this when he was here, maybe not. Basically, that mulch suppresses weeds, then there's no need for tillage. The problem is it's hard to generate enough of that cover crop to create a persistent season-long mulch that controls weeds all year long. It's also hard to ensure complete kill of the cover crop because after you roll it and crimp it, sometimes it'll pop back up. The time it takes to grow a high biomass cover crop reduces the amount of time you have for your cash crop, right? Especially in northern New England. And if you have a mixture of cover crops, it exacerbates all of those because there's no optimal time to kill that cover crop mixture, right? You're either waiting until hairy vetch is ready or you're rolling too early and the hairy vetch comes back. So it can be a big mess. So we wanted to ask, can we use plastic tarps after we roll these cover crops to ensure more complete kill? And would we get some additional benefits from those tarps? Because depending on the type of tarp, we might be minimizing light, we might be changing temperature, all of those things. So that's basically the idea behind this. We planted a rye hairy vetch mixture in last fall, and then we had 3 crimping dates, kind of an early date, June 1, a mid date, June 8, and then June 22 as our late date. Then immediately after crimping, we applied our tarp treatments. We had 3 treatments, black, clear, and then no tarp. Kind of looked like that. That's another picture of what it looked like. And then those tarps were removed, and we planted cabbage into those plots, transplant cabbage, on July 7. And you can see when we removed the tarps, there was a neat checkerboard. We had done something to that residue. We had done something to those plots. And you can see what it looked like pretty soon after transplanting. It looked pretty good. That's a nice weed suppressive mulch. We harvested cabbage September 14th, so we just got those data. At the same time, we measured a whole bunch of other things that will hopefully help us interpret those data, including real-time soil moisture. But there's just what it looked like just before we harvested. You can see things have gotten a little gnarly in there. Here's the whole reason I'm showing you this, because I got this imagery and I'm like, I got to show them this imagery, right? So this is from a drone. You can see checkerboard patterns. They're very cool, right? Here's the direction where we did our rolling. That was the whole plot factor, early, mid, and late, replicated 4 times, and then randomly assigned the tarp treatments. This is where the black tarp was, right? So these were taken not too long ago before harvest. And you can see, doesn't look too green in there, other than the cabbage. That's pretty cool. The clear tarp, a little more green, and a lot of that was crabgrass. Like, I think what we did is we created a greenhouse effect with that clear tarp and stimulated the crabgrass that was in the seed bank, and it popped up. And this is where we had no tarp, and you can see lots of green, and that was all hairy vetch that came back. Here's the yields of the cabbage, fresh off the presses, right? I just got these data like a couple of days ago. And I thought for sure there was going to be a tarp crimping time by tarp interaction, but there was not, even though these data look like there should be. There was only a tarp main effect. Basically, the black tarp had the highest yields. The clear and no tarp had indistinguishable yields, right here. And that's the story, right? The black tarp nearly doubled cabbage biomass compared to the clear and no tarp treatments. And that black tarp effect was independent of the time that the cover croppers rolled. However, I think if we had more reps, we would probably see an interaction. Very few weeds and no regrowth of cover crops in the black tarp, but we need to confirm that with our weed data. Lots of crabgrass in the clear, lots of hairy vetch in the no tarp treatment, need to confirm. But that no tarp treatment is what farmers do. That's what they do. So it looks like we can bump yields up in that system simply by placing a black tarp for a small period of time. All right, that was 112 slides. Thanks.
Questions?
There you go.
So in your first, in your first project, you sort of put in the quadrats on your stability versus performance or whatever it is on the x-axis. And you put the quadrats of high stability and high whatever. Those quadrats were sort of not meaningful to, so my suggestion is, why not transform the data to a z-axis where you've got a 0 center line, and maybe then your quadrats become more meaningful around 0, and maybe your treatments would fall out more.
Yes.
Something meaningful.
Yep, I like that idea. Yeah, completely arbitrary, just breaking the data in half on both axes. So you're exactly right, and that's certainly the next stage of analyses. And there's the other stage is there's actually a third trait that we had data on, and that's grazing tolerance. And so I need to include that trait range and then look at it relative to the total trait range, more in a regression. So there's a whole host of things that need to happen, and that's certainly one of them. That's a great idea.
I was curious, how soon did you put the tarp on? Did you do it right after the rolling?
Yep, immediately. So roll it and then immediately put the tarp on. So those treatments that got rolled earliest, because we had 3 rolling dates, they end up having a tarp longer than the other, because all the tarps came off at the same time, and then we planted right into that.
And how thick was the tarp?
Good question. The tarp, I don't know.
Plastic or was it tarp?
It was black plastic. Yeah, tarp's a bit of a misnomer. Yeah.
And in the tarp experiment, was there any irrigation or not?
Good question. There was irrigation right at the time of transplanting, just overhead. And I think maybe one more time. That was it. And there was 0 weeding, there was 0 anything. There was a little fertilizer applied at the time of planting as well, but that was it. And we have had the worst drought in recent memory this summer. And so it's pretty amazing how that worked. And so we have the water, the soil moisture data, so it's going to be really cool to look at that. But the interesting thing too is, like, when the tarp was on, we did get a couple of rain events, and the tarp basically excludes the water. So there's some interesting trade-offs, and I'm fascinated by this. Was kind of a spur of the moment study. It was a undergrad research project, and we all kind of pooled our resources to make this thing happen. And we're going to do it again at a couple different sites next year to really get some more data and maybe add a couple more treatments and really kind of understand that better. But it's something that farmers can take that information and. Yeah, you don't want to deal with a whole lot of plastic, but the plastic's not out in the field very long either, so it's not breaking down, so you could potentially recycle that plastic over 10 years or so. Probably more appropriate to small scale than large scale production. But if it works and we can demonstrate it works, then producers will figure out a way to scale that system up if they're interested. Yeah.
Why do you think it works? The soil temperature?
I don't know. I think I think I had initially thought that the black plastic was going to heat the soil up, but that's not the case. The soil was much warmer under the clear plastic.
So you measured.
We measured. Yeah, we've got the temperature data, and so I think it was probably a combination of things. I think that the black plastic killed the cover crop really well. I think the light. I bet a bunch of weed seedlings emerged, and they had no light, and so they basically, that was it. Whereas in the clear plastic, weed seedlings emerged. We warmed up the soils even more, and then when we took that plastic off, they were ready to go. They were getting light under there. We basically primed them to take advantage as soon as that plastic came off. That's my guess. Hopefully we can piece that out with the data that we do have, but that's another thing that we want to understand is what's the mechanism.
Do you think your organic growers will resist putting plastic in?
Well, that's why I was saying that. I think initially they might, but if they can recycle that plastic and use it over multiple seasons, because you're not leaving plastic out in the field all season, it's coming off after 2 weeks or so, 3 weeks. You can get a lot of use out of that. Great questions. Yeah.
So with your weed and your climate data, and I might have missed this, but clearly there's a connection there between climate variability and the kind of weeds you're going to get. But isn't sort of the bigger issue not necessarily that there are going to be shifts in weed species composition, but really maybe the amount of weed? And so did your data suggest any changes in that?
Yeah, we have those data, and we've looked at those are some of the response variables in some of those partial least squares regression models that I showed you. I didn't show you the results for that. I don't remember offhand what the results are, but we can look at that. So one of the things I do remember is that species richness. There was a relationship between the climate variables and species richness. I didn't show those data, but I'm in the middle of putting that paper together, and so hopefully in the next year or two, it'll all be there. But yeah.
Any other questions? All right, let's give them a
Thanks.